DNA replication. A replica refers to an identical copy of something. So you could have a replica of the painting. It won't be the original one of the artist, but it will be a copy of that particular painting. So when we talk of DNA replication, it is the process where a DNA molecule makes an identical copy of the original existing one. Why does DNA replication need to occur? It's very important because it ensures that the genetic code is passed to each of the daughter cells during the processes of cell division. If you look at this diagram, it's showing you on the left-hand side a cell with 2 chromosomes, but those chromosomes are referred to as being unreplicated chromosomes. So a chromosome like this would be referred to as being unreplicated. When it undergoes the process of replication, it then forms a chromosome like this one on the right hand side, the X-shaped one, which is made up of 2 chromatids joined by the centromere. These 2 chromatids are identical to each other because they are copies made by the process of replication. And the same thing for the one in the pink one. We find that this one also, as a result of replication, now we've got 2 chromatids joined by the centromere, which is known as a replicated chromosome. These cells eventually undergo some form of cell division, and if it's mitosis, it will form 2 cells, and these 2 cells will be genetically identical to each other. Each chromosome will separate during anaphase, and the centromere will split, and you'll have one unreplicated chromosome moving to each of the cells that are formed. So for the pink chromosome, we're going to find that the centromere will split, and each of these chromatids will separate, and you're going to have it now being called an unreplicated chromosome, one going to each cell. And the same thing happens for the blue chromosome. So we'll find that it's going to separate, and we have one unreplicated chromosome going to each cell in the end. So whatever information was in the original cell, it had a capital A and a small a, you'll find that the cells made by the process of mitosis will be genetically identical because they receive a copy each in each of the cells, and those copies of the genetic material were made during the process of DNA replication. When does DNA replication occur? It occurs during interphase. So if you know the cell cycle, the majority of the time the cell is within interphase. During interphase, the main things which occur is the growth of the cell and DNA replication, the copying of the DNA. And later, when it will go into cell division, either mitosis or meiosis, which has the different phases, prophase, metaphase, anaphase, telophase. And at the end of that, we're going to have new cells formed, and we just spoke about why it's very important that interphase needs to precede or come before the process of cell division. Where does it happen? It occurs where the DNA is found within the nucleus. Now, how does it happen? What is the process by which DNA replication occurs? So if we have a DNA molecule, like the one pictured here, you can see it's a double-stranded molecule, and it's twisted, it's a double helix. We'll find that the double-stranded molecule needs to unwind firstly, it untwists, and then it also unzips. So here you can see this enzyme, what it's doing is it's breaking the weak hydrogen bonds, and it's unzipped that molecule. Then complementary pairing occurs by free nucleotides from the nucleoplasm coming and joining in. So here we find, here's a DNA molecule, and you can see there's an enzyme that has unzipped it by breaking the weak hydrogen bonds, And now we have complementary pairing by free nitrogenous bases. If there's a T will join in there. If there's a C, we'll find a G will join in here. If there's a G, a C will join in on this side. And in that fashion, we've got free nucleotides joining in, and an enzyme will come and join the sugar phosphate backbone so that this new strand that's formed will occur on both sides. We say that both strands acted as templates, and in that way, what happens is that this complementary pairing is basically re-zipping on either side of the opened molecule. And then those two molecules of DNA that have formed, they will rewind and form two identical DNA molecules. And the entire process is controlled by enzymes. So just to recap and look at the process again, here we have a DNA molecule which has already unwinded. It's not twisted anymore in a helix. What will happen is after unwinding, we're going to find that this molecule will be unzipped where the weak hydrogen bonds will break. And now we find that the strands separate from each other. Right, so for the sake of comparison, we're going to keep the original molecule on the top. So we can see here it is what was on the left. TCAG will now be separated from these nucleotides on the right hand side, the AGTC, which now on the right hand side. Both of these strands, we say, act as templates for new nitrogenous bases to come and join. All right, so we've got free nucleotides. These are extra nucleotides that are floating around in the nucleoplasm. They will now come through and they will join on either side complementarily. So if there's a T here, we're going to need an adenine to join with the thymine. On the other side, if there's an A, you're going to have a T, a thymine joining. When we have a C, we know complementary pairing. C joins with G, cytosine and guanine. So here a G with a C, a with a T, T with a A on the other side, G with a C on this side, and a C with a G on this side. So what we've had here, this was the original strand on the right-hand side. This was the original strand on the left-hand side. And on the inside, we've got the new strand that was formed by complementary pairing. If we compare these two strands, we find that they are identical. TA and here's TA, CG, CG, AT, AT, GC, GC, and if we compare them with the one on the top, they are also identical. So these two identical strands would then rewind and form now two DNA molecules that are double helix. We know that this process of forming a copy can only occur because of the complementary pairing. And that's why the discovery of Watson and Crick was quite significant, because when you understand that DNA is made up of a double helix, which is made up of nucleotides and complementary pairing, then it makes it easier to understand now how this process of replication occurs, as well as how the process of protein synthesis occurs, which we'll get to in one of the future lessons.